Hi, all. Welcome back. We're going to continue talking about muscles, and the next couple lectures we'll talk about kind of the role that ATP plays, the role that different ions like calcium, sodium, phosphorus play, but also we're going to talk about the actual like components of how a muscle is relaxed and how a muscle is flexed. Okay, so first of all, it's a coupled reaction. Um, and so you get this contraction and excitation at the kind of the reverse of each other. And I'll show you what I'm talking about here in a second. Okay? So during relaxation, when the muscle fiber goes back to its original shape or somewhat its original shape, okay, calcium ions are stored back in the sarcoplasmic reticulum. Okay, so we talked about that last time. They're stored back and they get pumped back in. Okay, and this is an active thing, so it has to use energy to pump those calcium ions back into the SR. Troponin and tropomyosin move back to their original position or their relaxed state, okay, which means they cover the actin molecule binding sites. And we'll look at this here in a second. When the muscle is stimulated, okay, you get an impulse from a motor neuron. That impulse is going to stimulate the sarcoplasmic reticulum to release calcium ions into the cytosol, which is just a liquid in the cell. Those calcium ions will bind with troponin. Okay, so remember we have a troponin tropomyosin complex. They bind with troponin. When they do that, it changes the shape. Okay, and it changes the shape, so now that actin binding site is open. Okay, when that actin binding site is open, then the myosin head can bind to actin, okay, and that'll form a cross bridge. By doing so, then the fibers can be moved. Okay, the actin, the thin fiber, and the thick filament, they can be moved now because there's an attachment, there's a cross bridge that's formed. Okay, so let's look at a diagram of this. Okay, so here you can see troponin and tropomyosin, they are interacting with each other. When they interact with each other in kind of a resting state, those actin molecules, their binding sites are covered, okay? So nothing can attach to them. That's where calcium comes into play. Calcium can bind to troponin. When it does, it moves this molecule, okay? So the charge of calcium changes the shape of these two molecules. When that happens, okay, now these active sites are open, okay? And because those active sites are open, the myosin heads can now bind to them, okay? And they'll bind to them, and it uses ATP to do so, okay, to help with that binding process. But they bind up. That ATP is then broken up, okay? So the ADP plus inorganic phosphate is broken up, and that allows for the myosin head to move, okay? So it will change its shape from kind of this shape to this shape. So it's like it bends the head in one direction, flicks it, kind of thing, which we typically call a power stroke. Then a new ATP molecule will bind and break the connection and then move the head back into its original kind of formation. And then this happens again. Calcium, if there's plenty of calcium, then the filaments don't move back over to the binding site. Okay, but this can now attach to that binding site and move it again. Do a power stroke, and then attach, power stroke, attach, power stroke, and keep working that way extremely fast So the muscle fiber then will contract, and that's what causes our muscles to flex, and cause, depending on whether we're talking about concentric or eccentric or isometric motion. We'll get to these in a little bit. Okay? We'll determine whether we're actually moving, like your muscle, or if your muscle is stationary and just flexing, these kind of things. But this is the premise on how the muscle works. Okay, you have actin molecules or actin filaments binding with myosin filaments, and they do so because calcium changes the shape of the tropomyosin complex that's covering the active sites. Right, we call this a sliding filament model. Okay, and so when this occurs, sarcomeres shorten because the thick and thin filaments slide past each other. So they're not changing shape. The thick filament remains the same size, the thin filament remains the same size. They're just now sliding past each other, which changes the shape of the overall muscle and causes the muscle to contract. Okay. We talked about the H zones and the I bands. They narrow. Okay. The Z lines get closer together. That's where the thin filaments bind together, where they end up together. One thin filament and another thin filament. That's what we call the Z line. Again, thick and thin filaments do not change shape. Okay. And the overlap of the filaments just increases when this occurs. And I'll show you a photo of this. Okay? So here's relaxed state. You have thin filaments, actin-based filaments, thick filaments, myosin-based filaments. Okay, you have this zone. Remember, we were talking about the H zone, the A band, the Z lines, etc. We have these zones where there's no overlap. Right? And then we have zones where there is overlap. When the muscle contracts, the thin filaments move and the thick filaments move, okay, so that they overlap each other, and so that the bands get closer together. The Z lines get closer together, the A bands get closer together, the H zone is reduced, how much, or the lack of overlap is reduced, okay, and then they can contract further and get really close to each other and decrease the A band quite a bit. Now, relaxation occurs when the stimulation stops. Okay? And it's not just the stimulation stops, it has to do with the chemicals. It has to do with the ions that are present in the system. Okay? It also has to do with an enzyme. Okay? Acetylcholinesterase. Acetylcholinesterase, okay, huge word. It'd win you a lot of points in Scrabble if you had all those letters. The point with this, though, is acetylcholinesterase is an enzyme that consumes or decomposes acetylcholine. So when acetylcholine is released from the motor neuron, it's in the cleft. Okay? If there's no more response, if there's no more need for the muscle to respond, there's no more communication, there's no more action potential, then acetylcholinesterase will consume all the acetylcholine that's still in that cleft. So it makes it so there's no more synapse, there's no more action potential coming through. That will stop the impulse, okay? And then you'll have relaxation. The muscle will start to relax because there's no more impulse being sent. The sodium potassium channels will be closed. The calcium will start being pumped back in to the SR, and the muscle fiber will go back to its original state. Okay, so those calcium pumps are going to pack, again, they're going to pack that sarcoplasmic reticulum full of calcium. Right? They're pumps, they have to use energy to do this because the sarcoplasmic reticulum already has calcium in it, and so part of that diffusion osmosis situation is if you're going to keep sticking positive charged or negative charged molecules on one side of a membrane, you're going to have to use energy to do that. And so the calcium pump will do that. It pumps it in. The tropin and tropomyosin complex covers the binding sites again on actin, okay, and it just goes back into what we call the muscle fiber relaxation state. Okay? So myosin and actin can't bind together, and we kind of already talked about that. All right, let's talk a little bit about energy and the energy that's used for these contractions. First of all, ATP. We know this as the currency of the cell, okay? ATP is the energy for the cell. The reason why ATP is the energy of the cell is because it packs a lot of energy, but it's not super stable, okay? So ATP is adenosine triphosphate. Okay, it means you have three phosphates groups, three phosphate ions bound together. Well, the phosphate is a negative charge, and so you're binding three negatives together. They want to break apart. Okay? They're not very stable, but that breaking apart is going to release energy. So yes, we can store a tiny bit of ATP. So in your muscles, you will store some ATP. Okay? Not for very long, and it's normally just the first initial muscle contraction is the result of ATP reserves. We can't store for very long. Again, it it tends to break apart readily. Okay? Another option of short-term energy source, okay, is creatine. Some of you have probably heard of individuals who lift a lot of weights, trying to build muscle, taking creatine. Okay, creatine is a molecule that's going to hold a phosphate. And so as long as there's ATP in the system, creatine will carry a phosphate that allows for ATP to be regenerated. Okay, so you don't have to go through cellular respiration and regenerate it. It can regenerate ATP kind of in a closed system a little bit. Okay? That phosphate can be donated to drive work. Okay? So this is why people use it as a supplement. Okay? It gives you more energy to regions that need energy right away. And so this is why individuals typically use creatine to build muscle, is because if you're lifting lots of weight, okay, you need as much energy for those muscles as possible. Okay? The problem is creatine is also not a very good source of energy, and it really can only fuel about 10 seconds of intense muscle contractions. Okay, so tiny bit from ATP reserves, tiny bit from creatine phosphate. The majority comes from cellular respiration. Okay? And this is why we need good blood flow to the muscles. Okay? This is why a lot of athletes need to warm up first. One of the reasons, okay, if you warm up your muscles and you stretch, okay, you're allowing blood flow to get to those muscles. You're kind of slow rolling the muscles. You're not just going intense muscle workouts right away. You're doing a less intense muscle contraction, allowing for time to get blood to that region. That blood's carrying oxygen. Okay? That allows cellular respiration to work. This is going to generate ATP from glucose. We store glucose in our muscles as glycogen. So it's just a little more complex glucose molecule. It's bound a little differently, okay? And that's going to generate ATP, but you have to have oxygen in order for this to occur. And that's the cellular respiration part, okay? And so as long as you have oxygen in the system and energy to feed the muscles, then you can sustain intense workouts or intense muscle contractions for a very long period of time, because we can store a lot more glycogen than you could store ATP or creatine. You can store glucose for a long period of time inside your cells. Okay, let's look at the phases of cellular respiration. Some of you are fairly familiar with this, so we're going to just kind of cruise through them. And it's not super important, but it's nice to know where the energy is coming from, where are the pitfalls, and if you just get up and start sprinting right now, what's that burn? Okay, what's that feeling? Okay. The first phase of cellular respiration is anaerobic, is the anaerobic phase. And anaerobic means no oxygen. So no oxygen is needed and no oxygen is utilized. Okay. This is what we call glycolysis. This is breaking down glucose molecules without oxygen. Okay. Another word for this is fermentation. Okay. And it occurs in the cytoplasm of the cell. It's not associated with the mitochondria per se. It produces very little ATP, but it's an initial source of energy. So after the ATP reserve is gone, and if you haven't had very good blood flow yet, then this can take place. No oxygen's in the system. You can burn some ATP, or you can burn some glucose, generate a little bit of ATP, and then when the oxygen comes to the system, then you can go into the second phase, which is aerobic respiration. This means oxygen's involved. The products of the anaerobic phase fit into the aerobic phase. Okay, so glycolysis generates a molecule called pyruvate, and pyruvate is then converted into a molecule called acetyl-CoA, and that falls into the citric acid cycle, or the Krebs cycle, and they generate some molecules, some electron carriers that go into the electron transport system and generate a lot of ATP. Okay? The nice thing about our bodies is we can store some oxygen in our myoglobin. Okay? So we have hemoglobin in our blood that carries oxygen, and myoglobin in our muscles that store oxygen, a little bit at least, okay, to keep this process going. So let's look at kind of a diagram of it real quick. So glucose, which could be in the form of glycogen and then just broken down into smaller molecules of glucose, outside of the mitochondria, inside the cell still, it can be broken down through glycolysis, generate a little bit of ATP, just about 2. Okay? It's going to produce pyruvate or pyruvic acid. Okay? If there's no oxygen, that pyruvic acid or pyruvate will become lactic acid. This is what that burn is. Okay? If you just get up and start sprinting and your muscles feel like they're on fire, it's the result of lactic acid buildup. But pyruvic acid or pyruvate can then be converted into acetyl-CoA, and that can be used in the citric acid cycle. And the citric acid cycle itself doesn't generate much ATP, but the thing that it's not showing you here is it's generating electron carriers. 2 main electron carriers, NADH and FADH2, are generated, and they'll go into electron transport chain, which is just a fancy word for a bunch of proteins that generate ATP. Okay? And through the process of the electron transport chain, then we generate quite a bit of ATP. So for one glucose molecule, you're making about 28 ATP in the electron transport chain, 2 in the citric acid cycle, and about 2 in glycolysis. So a total of around 32, somewhere in there, depends on the individual kind of thing. All right, so let's talk about what happens when there is no oxygen, or when you have a supply of oxygen and then you lose the supply of oxygen. Okay, so first, anaerobic respiration, the formation of lactic acid, okay, will happen when there's no oxygen. Okay, so you'll have that fermentation process, you'll form lactic acid, okay, as kind of a byproduct of that. That lactic acid can be further reduced back to glucose when there's oxygen around. Okay, so it's not permanent. It's not like you produce this lactic acid and then you got to get it out of your body. As long as there's oxygen around, lactic acid can be switched back into glucose. So the process can utilize the oxygen. But our metabolism shifts from anaerobic to aerobic and aerobic to anaerobic, just depends on how much oxygen's in the system. How strenuous is the muscle activity? Did you allow for blood flow to get to that muscle and generate a good supply of oxygen? Now, if you have very strenuous muscle activity for a long period of time, there may not be enough oxygen and you may switch to an anaerobic respiration. So oxygen debt, okay, is going to be when you're lacking oxygen. You don't have enough oxygen. And so marathoners will typically have this happen. So they'll stretch and they'll do good, and for the first, it depends on the individual. Maybe for the first 10, 15 miles, okay, they're great, and everything is aerobic respiration. And then some of their muscles they're just not getting enough oxygen any longer, and all the myoglobin, the oxygen from all the myoglobin is used up. You're not getting enough oxygen into the body, and so portions of the muscle will start going into anaerobic respiration, and they'll start producing lactic acid, and that will fatigue the muscle, and that will accumulate in the muscle. Now it will regenerate into glucose after the muscle's done working, and that oxygen can resupply and be resupplied to that region. It will kind of generate it back, but nonetheless, you can have both ends. If you just start sprinting right now and you don't stretch, you're going to feel the lactic acid buildup. If you go for a very long period of time and you utilize all the oxygen in the system, you can build up a lactic acid response to that too. Okay, and so this is kind of how it works. Glycogen, again, just a more complex form of glucose. It can be reduced into glucose. Glucose then goes through glycolysis, produces pyruvic acid or pyruvate, which then becomes pyruvic acid. A little bit of ATP is generated. That pyruvic acid becomes lactic acid. Lactic acid, if there's oxygen, right? The liver can take that lactic acid and convert it right back to glucose, and you can just get a cycle going where that glucose can be utilized. So nonetheless, we do have options for oxygen debt, but buildup of lactic acid will happen. This is part of the fatigue. Okay? So muscle fatigue is the inability to contract a muscle, and this can happen in quite a few different ways. Okay? So first of all, you could have decreased blood flow. Okay? depends on like the region of the body. It depends on the temperature outside. It depends on the individual. But if you don't have enough blood flow to that region, you're not getting sufficient oxygen, you're going to fatigue those muscles. Those muscles will be going through anaerobic respiration, and then you'll get a lactic acid buildup. You'll utilize all the free oxygen in the system, and it just won't work anymore. You can get ion imbalances in the sarcolemma, which is the cytosol, which is the liquid portion of the cell. You can get an ion balance where you don't have enough sodium, you don't have enough potassium, there's not enough calcium in the system. These different ionic balances will determine whether or not the muscle, whether or not that actin myosin head reaction can work, or whether or not the channels will open for sodium and potassium to move and create that or allow for that action potential to stimulate the release of calcium. Of course, some of it can be mental, a loss of desire because of you're approaching fatigue, or just because it's not of interest anymore. And then, some individuals say it's controversial with lactic acid, but I think most of the research suggests that if you have an accumulation of lactic acid, it can fatigue the muscle. A muscle cramp is often result here in ion imbalance, okay, and electrolyte concentration imbalance due to either calcium, sodium, or potassium. Normally it's potassium. Sodium is, we have a lot of sodium in our diet typically, and so it's normally potassium that's lacking. But that can cause the channels to not be open, and you can get a cramp, or you can get situations where the calcium it never comes off the troponin, and then the muscle is contracted and it won't relax, and so depends on the type of cramp, but again, normally the result of ion imbalance. All right, twitch muscles. Okay, so twitch muscle is going to be a single fiber, single impulse. Okay, so motor neuron sends a signal to a fiber, the fiber contracts and then relaxes. Okay, there is a latent period. This is a delay period. Okay, so we can see this. It depends on the muscle fiber. It depends on the individual. Some people have very little latent periods. And you can do twitch muscle reactions. So normally there's a switchboard and there's a light, and when you see the light or hear a beep or anything like that, you push a button. Okay, and based on that reaction time, we can get an idea of what the latent period is in the muscle. Okay, there's other ways to do it, but that's one way to look at reaction time, is often how much latent period. It's part of the latent period. Again, it could be related to your eyes and your ears and other things, depending on the stimulation. Okay, period of contraction. Okay, so after the latent period, you have a contraction where the fibers are being pulled. Okay, and then you have a period of relaxation, okay, where the fibers are relaxing. Okay. Remember, during the relaxation period, calcium is being pumped back in to the sarcoplasmic reticulum. Okay. During the contraction period, calcium is being pumped out, okay, of the sarcoplasmic reticulum to react with the actin myosin head, or the actin and the myosin head can then attach. Okay, length and tension relationship. Okay, so there is a resting length of a muscle fiber, and that will give the greatest force. Okay, so everyone will have a kind of a resting length for your actin and myosin filaments. When they're at resting length, that gives them the most force. And so you might say, just this is optimal maybe for you, like this distance. Okay, that would give you the most force. If it's too short, the fibers can't contract all the way. You can't. There's no room to push the actin filament and slide it any further. If it's too far, okay, you also have an issue with that stretching motion in the sense that there's just less force. And the myosin heads, often if it's too far, they won't be able to bind to the actin molecules. Okay, so they'll miss the binding site. So maybe only a few of them would hit a binding site, and so the force is just, it's less. Okay? And so really it has to do, there is an optimal link. The heads have to be lined up with the actin molecules. If it's really short, there's no room for the fibers to slide. If it's really long, the heads are misaligned, and you get less force in both those cases. So you do have an optimal force. There's other ways at which we can stimulate a muscle, and you can have what we call summation. So this has to do with contraction and relaxation. If you have continuous contraction, okay, at known increments, but there's enough latent period and there's enough period for relaxation to occur, you'll get a pattern like this, where you stimulate, it produces a contraction, then it relaxes, then you stimulate, produces, etc. Okay? But if you have a partial tenuity or tetanus, a form of tetanus, you can have a stimulus. Before it completes the relaxation, if you stimulate it again, then it's starting at a different point, okay, and you can get more force, and then you can stimulate it again and get more force, okay, to a point, depends, okay, and so this really just limits the amount of relaxation between twitches. Okay? And this can be carried for quite a while. The other issue is when you have complete tetany, okay? And that's when you have so many stimuli that the muscle never goes into a relaxed state. Okay? And in some cases, you can have what we call tetanus, that is caused by, sometimes it's caused by ion imbalance, sometimes it's caused by a bacterial infection, but it will stimulate the nerves. There will be a continuous stimulation. It can be from the nerves, but it also could be that calcium won't come off of the binding site, and you just have constant stimulation. And sometimes you get what we call things like lockjaw, or your muscles can't relax. They won't relax at all. And that's a form of tetanus. We'll talk more about some of these situations after we cover the material. Okay? All right, and the last thing I want to talk about is the recruitment of motor units. Okay, so each motor unit is a neuron plus the muscle fiber it controls. And you could have one neuron, one muscle fiber. You could have one neuron, multiple muscle fibers. You could have multiple neurons, a few muscle fibers. So you can have different setup of motor units. Right? And it really depends on what the muscle is set up to do. If it's set up to do coarse movement, then you typically have a large number of fibers to a single motor unit. Okay? If it's a precise movement, you might have few muscle fibers and either more motor units or few muscle fibers per unit. Okay? And so often our muscles are set up to have some of each, okay? But it depends on the type of muscle, whether they have more coarse or more precise. Okay, so you can kind of see here you have two different motor units and they're attached up to the muscle fibers. And so often these motor units are going to have branched axons, which are going to attach up to different fibers. And so the number of fibers, it really determines whether it's going to be a coarse movement or a precise movement from that muscle. Now, when it comes to recruitment, okay, you can recruit more activity to a given region. Okay? And so I think most of us are fairly familiar with this, maybe not from a technical perspective, but from like a movement perspective. You can walk for a very long period of time. You can jog for probably less time than you can walk. You can run for less time than you can jog, and you can sprint for less time than you can run. Okay, this has to do with activating motor units. Okay, so a original stimulus will activate a certain number of motor units. If you need a more stimuli or more intense muscle movement, you're going to activate more motor units. Okay? And so you can continuously do this until you've activated all the motor units. And so like a full-on sprint, you've activated all the motor units, okay? And eventually you'll run out of energy in that region and you'll fatigue those muscles because every fiber is being stimulated.